Welcome back, students. Today's lecture is we'll be concentrating on genome sequence analysis and gene prediction methods. The previous class, we have spoken mainly on different aspects of genome sequence databases, biological databases, what is the necessity of a database, why should we use computational methods in terms of human capabilities and limitations, and the processing of the information of the huge genomic data we get up. So we talked about databases, but today we will take a little bit more on how we can just look into more details of the given sequence from a given genome, and specifically in terms of how you can identify what kind of genes the genome codes for, how many genes are there, what are the genes in terms of nature, structure, and what proteins it gives from the gene. But basically, we will talk more on genes and gene prediction methods. So the talk is divided into several sections. The first few sections are again introductory concepts. I will brush up a little bit quickly on this, then following up into a third concept, genome sequence analysis. Specific term we call that, we annotate the given genome. That's called genome annotation. So we annotate the genome to get more information out of it rather than simple ATGCs. And then gene prediction. How do you predict a gene from a given genome sequence, given a sequence stretch of sequences? There are many things, concepts of principal, features, and drawbacks for each gene prediction method. So there are many gene prediction method algorithms are available. Specifically in the syllabus, so they were wanted to talk about GRAIL, GENEMARK, and GLIMMER. So what I have done in the talk is that I will quickly introduce basic concepts of the how genes can be predicted, and then go into method by method, and then summarize at the end of the class. So if you are making notes for these two concepts, what are the drawbacks, features, and principles, so you can make a separate notes for this section. Hopefully I have made it some other information on that. So this talk was also given the time constraints, I was able to get help from some two other slide share presentations that was being referred accordingly. From that, I have taken some of the slides. Okay. So just coming back to the recap. So we have seen from past few classes what are the genomes, variability in the genome sizes, and we looked into genome sequencing methods and how it can be done. And overall, what one thing in commonality we can say that we just evolved from the lower organisms. There is a sequence homology. For example, a homologous gene in fly can be found in humans. In terms, we are saying that evolutionary conceptual manner, you are not different from the fly in some aspects. You are not different from monkey in some aspects. So the genomes are there. Genome consensus is there. And one more important thing is that we are going in a very unimaginable, fast-paced in terms of technological revolution, in terms of NGS and other. We are going not only from, we started in around 2010, 1,000 genomes, now we are under 10K, we may go into 100K soon. So there is a lot of data is coming out, and there is a lot of demand for bioinformatician data curators in genomics. So please keep in mind if you are looking for a field to work on in further your career options. So briefly on genome sequences, we have already studied in past few classes. We go through shotgun sequence method. The genome from genome is divided into chromosomes, or if you don't know the chromosomes of a given genome, you cut the genome into small pieces randomly, and then go into context scaffold. If you find a gaps, you go with traditional PCR methods to find the gap and fill. You make a complete genome map. So if you make a given genome, you make the maps. And it's as we already know that millions and billions of ATGCs are there in a given code, so you cannot fit it in a single book. If you are writing in a book of life, you need a multiple Bibles to write along. And again, if you go back and refer back to each from the stack, you take one book along, you go into specific sequences in that, you can match them to specific config. You look to previously assembled, you cross-check the assembly, therefore everything is fine. Basically, the genome sequence looks like a black and white to common man, some ATGCs. Even the sequencing methodologies, it will be in color-coded form. And the books are replaced by computers, right? You are not using books because physically it is impossible to store the information in the book form, and you cannot store it in a physical situation. So we use computational method. You use resources of data storage using computers. and then the books are replaced by folders. You may have a sequence given genome, you will have a sequence files that maybe you catalog on your own, and then you go to individual sequence file. You can see here 10 sequences are there. You are specifically focusing on one of the sequence here, and then the sequence can be cut up here. And the length of the sequence given in a file, 576 length. One more thing you need to remember, that although you are using computers, computers also have limitations. Do you think I can, in this given thing in the Windows space, do you think I can fit in 1 lakh nucleotides, 1 lakh ATGCs? I cannot. Again, you have to go with the computer limitations. Limited sequence you open at a time, you open at a specific length or limited length, because all the sequences are in FASTA format. Again, you analyze them given a chance. So by doing so, you take a small step and go into further analyzing. A single person cannot analyze, annotate a whole genome or analyze a whole genome. You need a consortium, you need a large group of individuals working together. And also you need to be an expert in that, what you are doing. If you are not expert, you don't know what you are doing, you may not get what you want. So let's look at random sequences. We just got it from Google. Let's take 30 seconds. I will count from now, second 1, and you just blankly look in the sequences. I have not demarcated it anything. You just look on the sequences. You can think of, based on, we are biologists, we are genomic, and we are specialists. You know about all these sequences compared to giving to my daughter, giving to my mother, who are non-specialists. They may decode something out of it, but I may decode something useful biological information. So let's take 15 seconds, look at it, the sequences. Think of the first thing what you can find it, and just write it down if you have a paper. I will get back in 15 seconds. 1. Okay. Whatever I thought, I have written. Actually, I took more than 15 seconds while preparing the class, so I might have more points. First thing what I have seen is that it seems like I can find A here, I can find A here, I can find A here, I can find A. So that's highly repetitive A's are here. If I take A, T together, it's again highly repetitive. I can find A, I can find repetitive. A, G, C is repetitive, mainly G, C, C, C repetitive. So I can simply blindly see that it's highly repetitive sequences are there. So does it make sense that the codon, the genome is whole highly repetitive? How it can code for specific gene? Why it's called book of life? That's a common man will ask a question. But as a specialist, you can decode something. You can have some conscious, and you can use that. Giving the yes highly repetitive, but I am seeing this GTGG or GGCC, a kind of 4 2 nucleotide, 2 nucleotide, or 4 8 nucleotide of the given sequence. Some commonality I can see, some motifs, something like kind of. Or I can see one thing, I know that start codons, what are stop codons. I know, see that I just quickly look into ATGs, I can find an ATGs here, or I can find a stop codons here. And if I assume this sequence, whatever given, is part of a gene, I assume from my basic understanding that there should be an exon and intron sequence here. And then I can distinguish them exons and introns based on whatever my fundamental knowledge. And if you take about the whole thing as a genome, you're looking at 3.2 billion base pairs, then you're looking simply in 2 concepts that are fundamentally. Whether some of this given a specific stretch of DNA codes for a protein or not. If not, it means a non-coding DNA, and if it's a protein coding DNA. And as a humans, we know it's a lot of junk, we will see. And also, the genes are divided into exons, proteins, or protein coding genes. So basically, given a stretch of DNA, if you don't know anything, you can look off all these parameters. And then you are looking at a specific window, specific length of the DNA here. And if given a time for you as a human, I want to analyze all these things for a given human genome of 3.2 billion, you cannot do. That's why you need a computational algorithms to analyze the data. And again, you need a group of people to work on that. Or you can take a specific concept in the given concepts here. You can work on one motif for whole your life and work as a bioinformatician. Only try to find the motifs in the given genome, and you take 100K genome or 1000 genome that are available. So coming on to fundamentals again, little bit more. And bioinformatics, a sequence motif is a nucleotide or amino acid sequence pattern that is widespread and has been proven or assumed to have a biological function. So you're looking at the sequence, whatever I spoke in previous slide, you're looking for a sequence motif with a nucleotide or amino acid sequence patterns that are widespread in a given sequence. And they have shown that they have some biological functional significance because you tested it in the wet lab. You have done experiments in the biological system. You have shown the importance of the sequence. If you take a DNA motif, DNA motifs are short, recurring pattern of DNA that are presumed to have a biological function. Mainly these motifs you call in terms of promoter region, sequence-specific binding of transcription factors to the DNA sequence. That's based on the motif recognition. And motif, if in a protein, a chain-like biological molecule is protein, and structural motif is a super secondary structure which appears in a variety of molecules. The motif, protein motif may be consistently common in some of the organisms. One more important thing we need to work on here is a putative gene and consensus sequences. It's also really important. So a putative gene is a segment of DNA that is believed to be a gene. Actually, it's a gene, nothing but. Putative genes can share sequence similarity to already characterized genes and thus can have a common similarity or homology. So for example, I'm looking for a C. elegans, fly, worm, mouse or bacteria or some other genes. I'm looking for specifically some part of the gene, for example. And then I found out that the gene will have specific sequences common to me, common to humans, common to flies. So then I conclude from the sequence homology that, yes, the gene, for example, you have seen Hox genes, right? They are pattern formation, anterior, posterior pattern formation. The whatever that makes head of the fly, whatever that makes tail of the fly, insect, are doing the same thing in humans also. So basically you can decode based on the sequence similarity and functionality. They can be called as putative genes. And what is the consensus sequences? In molecular biology and bioinformatics, the consensus sequence is the calculated order of most frequent residues, either nucleotide or amino acid. You are looking at the FASTA here, but if you go into amino acid sequence, you get amino acid codes here. They may be conserved again. Found at each position a single alignment. For example, you simply look here. So consensus sequence is the calculated order of most frequent residues, either amino acid or nucleotide residues, found at each position in a sequence alignment. Basically nothing but you have seen the motif consensus, right, in a sequencing in NGS. For example, here sequence 1, sequence 2, sequence 3, 4, 5. I have 5 sequences. I just blasted them, blasted nucleotide, blast 10. Then I found that leaving the first nucleotide in the order, all the remaining, there can be a kind of proportionally, there may be a consensus I can generate. There may be some variability, but in this position, out of the 5, 4 are T's. So I can say approximately it may be a consensus. Like that here T, all the 5 out of 5 T's are consensus. You can deduce some sequence, again test it. And here you're looking for a kind of consensus sequence provided here. Specific group of genes here, they may be putative genes. Location, you're looking -20, start of the transcription first, and 20 below, 20 upstream of the transcription start site. You're seeing nothing but ATGs here, consensus. So you can sequence, conclude that this is the sequence of that. The start codons are consensus sequences here. There is a amount of conservation among these genes. There is some variability again, but there is a, you see here also GTG. There is one variability. So you deduce the information in terms of the given sequences. So putative genes, consensus gene sequence important. So again, you look into all this in terms of the books. You go into look in the folders. You're looking in this folder. You can go either convert this sequence into complementarity in the opposite direction, or you can go for whatever gaps that annotation gives you. You can remove the gaps. You can realign it. You can again make a new file and give a name. You can go into plot, alignment, evolutionary, check consensus. You put an output in the protein output you want. You can go ahead. So quickly again, basics of a little bit more, because you need to in terms of gene prediction method, whatever we have spoken previously, you need to brush a little bit more in terms of prokaryotes and eukaryotes. Nothing else. You go quickly now. Different region, structural region, important signals of a given gene which is undergoing transcription or translation. We start with the 5' UTR here. It's already an mRNA here. 3' UTR, and the polyadenylation site. We go transcription start site here. DNA region promoter, and introns are here, and then exons are here. And then one more thing we call splice region, we will see into that, and the codons are here. Just have a little bit brush up your memory looking into this. You come up into different portions of the genes, and I say that these genes can be conserved, these portions can be conserved among organisms. Or given a DNA, can I found a different stretches of in the genome, I can found this transcription start site, can I found start codon, can I found poly A signal, and another splicing regions can I found or not. Again, coming on to bacteria and eukaryotes, prokaryotes and eukaryotes, they are fundamentally different. If you are using a specific gene prediction algorithm, you might have to use different parameters for each. For example, prokaryote, the translation, transcription are combined in the cytoplasm, but in eukaryotes, you have a distinction of compartments, and then mRNA, processed mRNA should transport to be in the cytoplasm, then ribosomes can access them. One more important criteria here is that, so basically the mRNA is here, mRNA here is fundamentally different. This mRNA will have a signal peptide or protein sequence that will help it is to export it into from nucleus to cytoplasm. So you can use these export signals also in the DNAs and do algorithm for predicting the genes, but you may not use same algorithm for prokaryote. Nucleocytoplasmic, can I use these export signals? Can I find it in the genome? Yes, you can find, because they are consensus sequences given mRNA molecule. One more fundamental difference in terms of eukaryotes, there are histones will come up. There is a processing, compartmentalization, there is repeated patterns, how the histones will align to given a specific distance of DNA. And prokaryotes have no introns. Promoter regions also vary from prokaryote to eukaryote. And also in terms of codon usage, humans, what codons we use, there is some difference in terms of the codon usage also, even the prokaryotes. Accordingly, the gene prediction algorithms changes. Let's take the sequence and again look into the consensus sequence. One concept at a time. I can find the consensus here in terms of the ATGs, in terms of the promoter binding, in terms of the start region of the promoter. Yes, I can deduce something out of this. Let's talk a little bit more, few slides on open reading frames, ORFs, given a sequence here. So if you find an ATG here, you can find a stop codon somewhere. We have spoken in the first class, I think, an open reading frame is a continuous stretch where a ribosome can start and then complete its translation given a stretch of DNA. So it usually starts from ATG sequence. If you zoom in here, these are introns, these are exons, then starts from ATG and then go into a kind of find a stop codon and it stop the translation process. A continuous stretch of DNA starting with one codon to the other last stop codon is called open reading frame. And also we know that the codons are triplets, right? They have triplets, three nucleotides at a time. We know that Obel hypothesis, triplet codons, fundamental molecular biology. But if given a sequence, you think of how many open reading frames you can find in a given stretch of DNA. One question you can ask. You're looking at a DNA here, 5', 3', opposite 5', 3'. Let's say you have a starting ATG here, it's okay. But how do the ribosome knows that it has to go with only start from here, take only these three? We don't know that yet. It's random. But if you take an example of a codon starting from here, triplets, then you take ATG here, you divide them into, and then specific amino acids will come up. But again, it can be taken from T also. You take from TGA, it moves along again, shifting one frame shift, and it can be taken from second, third frame, GAC, it can move along. Same way in the opposite strand, ATC, AAT, TEA, you're starting from CTA here, TAA, and then AA here. It can happen any of these forms, but again, you remember one more thing is that when the replication happens, replication is bidirectional. It can be going to 5', 3', 3', 5'. For each of the strand, there will be a template, and the replication fork moves along bidirectionally. It synthesizes both strands. And replication and transcription are simultaneous. So the transcription can also start this side or this side. A gene can be transcribed in this region, gene X, gene Y can be transcribed in the opposite region. So if the ribosome is coming along, it just looks for 6 possible reading frames. You look in the next slide. So for example, given a stretch of DNA again here, you are moving in this direction, 5' 3' direction. You look in starting from ATG, nucleotides are coming, stop codon is here. You move 1 frame, nucleotides are there. But you see coming from these 3 frames, the stop codons here, the red ones are stop codons. In this stretch, there is a long length. It's okay enough given the frame. But in the second frame, there is again stop codons are coming in between. In third strain, there is a complex here. Again, move in the opposite direction, reverse direction, you may have ATGs here. Some may not have ATGs at all. Some may not have stop and start codons at all. So basically, given a frame, there will be frames in the top, frames in the bottom. Total 6 frames will be there, and the top frames in this going in the direction are called frame 1, frame 2, frame 3. Going in this direction, frame -1, -2, and -2. Okay, 6 reading frames are possible. Accordingly, it goes. And then in general, the ribosomes or ORFs, the fundamental principle is that you will take a complete stretch without the stop codons, longest stretch of the DNA that takes from stop and takes into that given a open reading frame or given a stretch of DNA. Okay, you just remember the rule. In this case, the ribosomes at the frame is starting from F1 because it is the best possible combination given in the scenario. So that's like you read the previous slides and you look in the sequence now, you think of ORFs. How many are there? Again, in terms of how do you reverse the sequence, if you are moving in this direction strand, you are moving in the opposite direction, the softwares will help you which strand you are looking up and which frame you are looking in turn. If you are moving that way, the frame is positive, moving down, frame is negative. So when you talk about a open reading frame, you are taking both introns and exonic regions. For example, this is a stretch of DNA. This is not an mRNA. So you are looking at the DNA stretch here. So you have exon and introns. So why there are stop codons in this frame? Because you are looking probably a region of introns here, not the exons here. So the introns always cause a problem. If you have a code here, exon 1, exon 2, the amino acid sequence for specific gene and protein is combination. After Q, W should come. But there is DNA is there in between intronic sequences. There may be a stop codon. There is a premature stop of this translation. That's why splicing will happen. Exon 1, exon 2 will give continuously. Then it gives a process having the gene expression normal. We already know that in terms of the splicing mechanism, there is a 5' 3' region. There is a consensus sequence given in the exon intron bridge. You always have a GT here. You always have the AG here. Every intron exon sequence will have it. That's called as consensus sequences. Donor splice, acceptor splice regions are there. This is part of the eukaryotic mechanism, splicing. Based on these sequences, you can distinguish where is my intron, where is my exon in the given sequence. So based on now, looking in the splicing, looking in intron, exon, and ORFs, I can say how many splice sites are in there in given sequence, how many introns and exons I can distinguish. So given a software, you give the software some details, give the specific length of the DNAs. It can go along. It can give a specific some length it will take in between. It will show wherever the GT highlight, it show blown up. AG is there blow up. Then you can see that how many ATGs in splice sites are there in a given sequence. You can use based on that. So thus it brings to the little bit fundamentally better understanding of the concepts what we discussed. And based on those fundamental aspects of having, you are looking at the ATGs differently now based on the ORF, exons and introns, other splice. You can also use transcription start sites, polyadenylation sites. So you can look the genome in differently and you can, using those, you can annotate the genome. That's why genome annotation is called obtaining biological information from unprocessed sequence data, simple ATGs. It may be structural, it may be functional, depending on what you are trying to find. So raw genomic sequences can be annotated by either you have already an established set of cloned genes, for example, Hox genes, or expressed sequencing tags. You already have a library. We will talk this next class. You may compare them. You may annotate. You can annotate them accordingly, or you can go into gene prediction method based on promoters, splice site, polyadenylation, ORF, or you can use the softwares or algorithms. But that will be the class for next half of the portion, gene identification. So you know that prokaryotes, eukaryotes fundamentally differ. Given a prokaryotic genome, there is 85% of the genome codes for genes, protein coding genes. Surprisingly, for human genome, it is only 3 to 5% of the genome holds. And then in the given human genome, typical exon is 150 base pairs, intron is several kilobases. A complete gene mix introns and it's 100 kbs of distance will be there. So based on that, you need to use computational methods, ORF finders. You need to annotate each individual gene on your own. So only 2 to 3% or 3 to 5% basically. And you're talking about a lot of junk. We have lots and lots of junk DNA. So for example, if any software also, it is very happy to take a prokaryotic genome. There is not much complexity. I can easily annotate that. Don't give me any human genome, because it takes a lot of effort for the computers also go into their own algorithms. It takes a lot of effort to find something. And if you know right now also, the whole draft of the human genome still has some gaps. Maybe 50 or 500 gaps are there. We are still not able to fill the gaps because a lot of repetitive DNA, a lot of junk DNA, whatever possible assembles. Maybe in future one more AI will come, artificial intelligence, we may fill the gaps. We looked into the ORF prediction, some more text given here, taking 6 possible protein sequences, longest frame can be taken. It is easy to find the beginning. Finding the ends of ORF is easier than finding the beginnings. Maybe it's okay, but if you look in the beginning, you look for a start codon ATG, you look for a specific sequence upstream of ATG, start codon, some islands are there, CPG islands, that will help you to identify that. It's a first step, and you look into little bit notes, whatever I have spoken. Methionine is the first codon, but it changes again species to species. Given a ORF finder, a software, you use it, you have this kind of a given sequence, you give in FASTA format, and then it gives whatever ORFs are present. It gives specific length of the amino acids, whatever start and stop codons are here, if you zoom in. But if that may be true consensus with the previous known data or not, it may be not possible to give the right thing. So the computer, you can go for alternative initiation codon, you change the frames, then you again get into the point where you have a consensus sequence, or you know that this frame is good enough. So this is a GENEMARK results, one of the software that uses this. For example, you go with the length of the given ORF here on the direct strand, indirect strand in the negative. You have a coding frame 3 here. It takes specific gene of the bacteria Streptococcus, and then it gives specific length of the ORFs coming in that. Different ORFs, frame 1, frame 3. Score also it will give. Accordingly, you can generate different ORFs. This is again GENEMARK, one more algorithm that helps you in that. How do you predict is a true ORF? You can read a little bit more. You can compare it with amino acid sequences. And some of the problems in the ORF is that if the genome was not properly annotated, a single character sequencing error from the machine also can shift the frames, framing that you get inaccurate information of the ORFs. Then short exons are also overlooked. Multiple transcripts coming from multiple ORFs sometimes confuses the results. This is the limitations you can go into. One more important, whatever genes you find using ORF and other algorithms, they are species specific. Either you are looking at the bacteria, you are looking at the C. elegans, nematodes alone, you are comparing with humans. There is a gene species specific gene findings can be done. For example, common repeat sequence are species specific. Gene finding programs rely on information, basically on coding regions also. So whatever ORF we spoke briefly now, they are pattern-based gene findings. Excuse me. Mobile lamp. Go, first go. Okay, I'm back. So pattern based, you're looking at the promoter sequences, you're looking at characterization binding sites. There is a pattern can be established that can be statistically analyzed. But what about softwares? Softwares, there are a lot of algorithms that can help in predicting. And ORFs, it takes a lot of time to find ORFs and go and analyze them. But if you take a software, there is a speed in the annotation process. But one more disadvantage is that softwares also, in terms of errors prone, you may not get accurate information also, but it's 90% you can confident. And then there are two strategies: homology searching, ab initio prediction based on the softwares. So this is again, the information looks like that in the given sequences. The software you can put it. And one more example we will see in that. Again, one more information is that there will be again limitation in terms of the window. 100 base pairs length only some softwares can allow to find whatever you want. So this is a little bit more, that's what I'm saying, 100 base pairs long. You can read it a little bit more points on standard content-based alignment procedures. But given time, I will quickly jump to GRAIL and GENEMARK, most of the sequence information that will help you to understand. So most are web-based. For example, submit a sequence, you input it. The length may be limited, as I suggested. You may not put 1000 base pairs at a time. You have to select the parameters, interpret the results, and then based on the generation of the software, you may get a graphical version or non-graphical version of the output you are looking for. For example, if you take ab initio prediction method, there are some algorithms that used. That's called GRAIL, Gene Finder, Gene Scan, and some other models are here. So one of the thing is called GRAIL, that's based on neural networks. It predicts exons, genes, promoters, CPG islands, few other repetitive elements. And the features of them are dependent on prokaryote, eukaryote. Again, few programs are different in the base composition also, it changes. Mainly the accuracy is 100%. They are not 100% per se. There is accuracy problem. What are the errors in the sequence are incorrect calling of exon boundaries, exon intron boundaries, missed exons, failure to detect entire gene, sometimes it happens. So you need to use more than one algorithm to come to a conclusion that my sequence is consistently, this is the gene sequence for my given gene I'm looking for. One more thing is called homology searching. Finding genes are long sequences based on whatever transcribed, for example, cDNA and ESTs of a given gene. You blast specific given genes with a genome, or you blast 2 genomes at a time through BLAST10, BLAST-P, or BLAST-X. Then it gives a specific consensus sequence based on the homology also. You can deduce a gene, Hox gene from C. elegans. You blast it with a human, whatever catches up, those you can take and start working on that nucleotide sequence, spacing and a specific oxygen. So whether homology is important, ab initio is important, each one will have limitations. Still, there are more advanced programs are available. GeneI, GeneView, Generation, GeneID, a lot of stuff is available, but try to concentrate on only GRAIL and Glimmer. For example, GRAIL is a gene finder for human, mouse, Arabidopsis, Drosophila, and E. coli. It works on based on neural networks, pattern-based recognition. Accuracy is 75 to 90%, 95%. Glimmer, Glimmer is specific to bacteria and archibacterial genomes. And this uses neural network. This uses a Markov model that you can look into that. What are the models? We'll briefly see. And it predicts 98% accuracy. How the Glimmer looks, it looks like this. You submit a sequence, you look for whatever features you're looking for. Next, one more is GeneMark and GeneScan. Look into that. It's for humans and vertebrate species. As I said, there are species-specific algorithms. This for bacterial and again archibacterial. This based on again Markov model. Accuracy you can combine with that. So GeneMark look like this. You can go into that. One of the ORFs given, you can look into deep into that. So we spoke briefly on different regions of genome, different regions of the given ATG sequences. how you can identify based on landmarks, based on the promoters, based on transcription and exons, and how you can use these computer algorithms to reduce your burden of physical labor to working on that. So you just go back again, look more detail into that, but I will quickly give you from scratch now. For example, I am a dummy. I don't know how to do gene prediction or gene annotation. How myself or someone of you, how you can start. So you have to start from whatever we have discussed in the previous slides. You start with finding a strategy. So choose the appropriate type of gene finder you want to work on. Make sure you are working prokaryote, eukaryote, it changes accordingly. And then you pick one. If there is no organism specific finder, at least you can make that what the output you want, make some sense out of it, make some hypothesis, accordingly use the benefit salient features of the algorithm, use the best one. How the algorithm work? The GRAIL is a neural network or topology based algorithm. It's statistical method, brush up your statistics. Given the ATGCs, it will have a network of common nucleotide there. It will divide the given set of data into training set, test set. The training set, it will again test, training set will be going into multiple round of algorithm for scrutiny. Then once it knows that training set is good enough, it compares with the test set. Then it makes some assumptions out of it. It gives the data in a kind of score. There's still again caveats in that. The net only performs as well as the training set. How the training set has been trained, that gives only the result in that limitation. There is a few things you can change. You cannot change much again here. You have to follow the algorithm, what it says. You can improve it in time, making the system more robust, more experience can given to the system. You can go in that. And again, Glimmer uses Markov model. Markov model is nothing but there is multiple states of the data. It will not take a previous step. It will take each step in the data analysis independently. It's just like rolling a die. You have 6 possibilities, but the first possibility is not linked to the second. You may get 1 every 6 times. You may get 6 every 3 times. It goes through probabilities. Hidden Markov model, read it a little bit more. How it is used to testing that in terms of E. coli genome. You can look into that little bit more details here. And one more importantly, each algorithm will have a parameter. So either it's a true positive, true negative, false negative. Based on that, algorithm specificity, sensitivity, correlation coefficient can be generated. A score will be generated for each algorithm. The best score, you are looking for an exon prediction. You can pick that method. You can use that method. Okay. So we'll meet again on Saturday. Thank you.